Welcome to the NWA ETC Project Echo. I'm Kent Unruh. I'd like to turn it over to our medical director, Dr. Brian Wood, to introduce our guest.
Great. Big thanks to Bob Harrington for doing this talk. Should be very, very interesting. You all should probably know Bob. Bob's a professor of medicine here and director of the Madison Clinic, and I will turn it over to him.
Great. Thank you, Brian. This noon we're going to talk about HIV cure, and a bunch of us just got back from the HIV conference, the CROI conference, the conference for retroviruses and opportunistic infections that's held in North America every year. That's kind of the big HIV conference. And there was a fair amount of data and presentations on a cure. One kind of exciting thing that there might be a second person who is seemingly cured of HIV. So it's a relevant topic, and we'll try to march through what's known, and then I'll included some of the things that were presented at this CROI conference just yesterday. Just in talking about cure, this is the outline of what I'll review this noon. Why do we want to cure people of HIV now that we have good therapies and it's pretty well controlled for most patients? What is the HIV reservoir? Why is it difficult to cure people, and where is HIV hiding? What about the effect of antiretroviral therapy and initial predictions years ago on what it's going to take to cure people from HIV? We'll talk about the difference between a sterilizing and a functional cure. And then what did the two people who have been cured of HIV, Timothy Brown, the Berlin patient, we'll review his case, and this new case of this baby from Mississippi teach us about the potential for curing all people from HIV. And then we'll talk about some of the strategies, activating and purging the reservoir, therapeutic vaccination, transplantation, that's stem cell transplantation, and gene therapy. So first, just by way of background, why do we want to cure HIV? I mean, we've got treatments now that are pretty good. You can take one pill once a day, and if you don't have any side effects from it, then it's kind of better than having hypertension. But there are some problems that remain. First of all, these medications are very expensive, and you have to be perfectly adherent to them for the rest of your life. Despite good control of HIV, the life expectancy is still lower than that of the general population. There's one study that's referenced here with a 50% lower chance of surviving to age 70 compared to the general population if you have HIV. And then there are toxicities associated with the medications, lipodystrophy, hyperlipidemia, renal and bone disease, as well as higher risks for vascular disease, liver disease due to co-infections with hep C, malignancies, even in people who are well controlled with their HIV. And then, as important, the stigma of being HIV infected is still crippling. It's tragic and true, and it contributes to social isolation, depression, substance abuse, and it is ruinous for many people's sexual health and their reproductive future. So wouldn't it be great if, rather than just having well-controlled HIV, you didn't have HIV? So this is just a schematic that looks at what happens to people once they get put on antiretroviral therapy. So with primary infection, your viral load goes to very high levels, and then it's controlled at a set point here. When you get started on antiretroviral therapy in this time period, your viral load drops precipitously. The first part of that viral load drop is mediated by really the death of actively or activated CD4 cells, and they have a short half-life of only half a day, and the kinetics of that are kind of this rapid decline in viremia here. And then there's a second minor population of cells that live a little bit longer, 2 weeks or so, and they decline, and the viral load declines from these cells at a slower decay rate. And then there's a third phase here that really represents the latent reservoir. Those are cells that have a very long half-life, on the order of years, or sometimes even longer, maybe decades, and those are the cells where HIV survives and persists, and that is what defines the latent reservoir. So how the heck do these cells get infected? Well, and how do they compose the reservoir? Well, it's really an accident. This is what happens with HIV infection of an activated CD4 cell. The virus enters the cell, it's activated, reverse transcription happens, the genome is integrated into the host genome, and then those activated CD4 cells will make a lot of progeny virus that bud off from its surface here, and because that T cell is activated, then it is usually killed. It either dies by apoptosis because it's been activated, or the immune system recognizes that it has HIV proteins on its surface and it kills it. But the fate of these activated CD4 cells is usually death. But every once in a while, maybe around 1 in a million, these cells will turn off. They will become deactivated, they will become latent, they will become quiet, and they have an integrated provirus in them that just sits there and will exist indefinitely for as long as that cell exists. And that is the reservoir. These numbers in an infected person are around 1 million of these central memory or latently infected T cells that are hanging out there. There was some data presented at the conference that suggests that it may be more than just central memory T cells that compose this latent pool. It may include cells that are stem cells, and they are marked by this CD133 positive. There are these other T cell subsets called gamma delta T cells. Some data that macrophages, glial cells, which are brain macrophages, and neurons may also be latently infected with HIV. But this is all kind of controversial and new data. And the anatomic sites where these cells hang out, they're kind of concentrated in lymph node, gall tissue, which is gastrointestinal lymph node tissue, spleen, and brain. So that's what the reservoir is. One question is, what's going on in the reservoir? Is there replication going on in these cells? In other words, are these cells occasionally producing virus that then is going through the entire life cycle and replenishing the reservoir with new cells that would have an integrated, that's the little red provirus there, that would have an integrated provirus in it? Is it replenishing itself like this? If this were the case, you'd expect there to be some viral evolution that's happening because there's some selective pressure and you'd have different sequences of viruses over time. The other option is that there is some residual viremia that's happening, meaning that a latently infected cell like this would activate over time for a variety of reasons. It may kick out some virus, okay? But because people are on antiretroviral therapy, either with reverse transcriptase inhibitors or integrase inhibitors, they don't really make new cells that are part of the resting reservoir. So it's not replenishing. It's just leaking out of the reservoir and not leading to any newly infected cells. Most of the data suggests that B is correct because there is limited evolution of the reservoir over time, meaning that there's not new viral sequences that are observed in people over many years. And if it was A, you'd also expect that if you intensified treatment for people, put them on 4 or 5 drugs, that you'd stop seeing residual replication. You would stop the cycle, the life cycle of HIV. And when we intensify people with raltegravir or other therapies, we often see little or no effect on this residual, very low-level viremia that's occasionally observed in people that is thought to come from the reservoir. So it's probably B, that the reservoir is not replenishing. It's just very long-lived and tough to get rid of. Now, Bob Silicano from Hopkins presented some very interesting data from people who are on antiretroviral therapy and looked at cells that were harboring provirus from these people that made up the reservoir, and then pulled out the individual proviruses from cells in these individuals and found and presented that 50% of the HIV provirus that's in these cells has huge deletions in it. So it's totally non-functional, just garbage DNA that is not doing anything. 28% of the proviruses that he pulled out were hypermutated. So these are proviruses that had not big deletions, but had dozens of mutations in them, rendering them replication incompetent. And another 10% had a variety of other mutations that were less dramatic. So that only 12% of all those proviruses that are integrated into the genome of these latently infected T cells was inducible and could produce replication-competent HIV. So it just speaks to when we're measuring the reservoir with DNA assays, what are we really looking at if only around 10% of what we find is really capable of reactivating and causing new viral infections? Something to think about. So what's going on in these latently infected T cells that make up the reservoir? Well, the answer is not much. The HIV provirus is crowded by these deacetylated histones. So those are seen here. These are these little gray balls that kind of bunch up the DNA. The provirus here is this multicolored linear structure, and it's all crowded in by these histones that prevent the transcription machinery from replicating that provirus. So histones are getting in the way of HIV growing. And then there's transcription factors, NF-kappa B and NF-AT, elongation factors that are sequestered or are in limited supply. Here in this diagram here, these transcription factors and elongation factors are not really accessible to the provirus, and so another reason why they're not making new virions. And then there's methylation of the DNA that gets in the way of transcription, and then some post-transcriptional blocks as well for RNA transport out of the nucleus and then translation into protein. So there's lots of nothing going on in the cells, and it's due to the fact that they have all of these changes that are preventing growth or transcription of the provirus. So in curing HIV, the initial predictions were that we were going to get rid of HIV in a few years if we had good suppressive heart for individuals that shut down viral replication. But it all depends upon the life of the cells that are infected. So going back to that graph I showed earlier, if all the cells that HIV lived in had a half-life that was measured in days, so that's the first order kinetics, then you would cure somebody from HIV in days to weeks. On the other hand, if they had a longer half-life, which is that second order of cells that lives for maybe a few weeks, but even so, if that's the only cells that were infected with HIV, then if you kept people on antiretroviral therapy long enough, then you would cure somebody from HIV in months to years. But the problem is we have these long-lived cells that have half-lives that measure in years or longer, and so the time for, if we just had HIV treatments, just antiretroviral therapy, these cells are going to stick around for a long time, maybe as long as 60 years, and that's the problem. That's why heart therapy alone is inadequate for curing HIV. So if we're talking about cure, it's important to have two, or it's not important, but there are two definitions. One is a sterilizing cure, meaning that we completely eradicate all replication-competent forms of HIV. So the reservoir is gone. There is no full-length proviral genome that is in a cell that could be reactivated. Functional cure is different, and in that case the reservoir remains, but there's permanent control of replication without antiretroviral therapy, and that, for example, would be elite controllers who just have a robust and controlling immune response so that they don't need antiretroviral therapy. So has HIV infection been cured? Well, there now seems to be two examples of that. The first was several years ago and was reported in the New England Journal, and that is the case of Timothy Brown, the Berlin patient. This individual had AML, acute myelogenous leukemia, and he underwent a bone marrow transplant. He actually had two transplants because his AML relapsed. But he was treated with TBI and intense chemotherapy twice. On this graph here, this is where he got TBI and chemotherapy to treat his leukemia. His leukemia relapsed, and then he got TBI and chemotherapy again. So pretty potent drugs to eliminate all his lymphocytes, which is where HIV lives. And then he was rescued with a bone marrow from a donor who was CCR5 minus. So in this case, he was given bone marrow cells, or cells that do not express CCR5 and therefore are resistant to HIV strains that utilize CCR5 to get into the cells. And then finally, because the bone marrow or the stem cells came from a different donor, those donor cells attacked any lingering cells that he had that were not only the source of his leukemia, but also were the source of his HIV reservoir. So the question is, for this patient, what is it that cured Timothy Brown of his HIV? Was it the intense chemo that was lymphocyte depleting? Was it the fact that he got a resistant marrow from a donor? Or was it the fact that his new immune system attacked any residual HIV that he had? And the answer to that question is nobody knows, but we're trying to figure out so that we could try and replicate what happened to Timothy Brown to some other people. The second person was presented at the CROI conference just a few days ago, and I've termed her the Mississippi Miracle. This is young infant who was born to a mother who was HIV without knowing it. The mother was a pretty long-term non-progressor, we think. Her T cells were high, her viral load was low. She was not on any HIV therapy at all, and she delivered at 35 weeks by vaginal delivery, a healthy young daughter. And she got no antiretroviral therapy during delivery, maybe because they found out at only the last minute that she was HIV positive. The child was quickly discovered to be HIV positive and transferred over to the University of Mississippi, where they tested her HIV DNA on her blood cells, and that was positive. They tested her twice, and also tested HIV RNA in her serum, and that was positive at 20,000 copies. And she was started on combination therapy of AZT3TC and nevirapine that 7 days later was switched over to Kaletra. And she stayed on that, and by day 20, so 3 weeks later, the child's HIV RNA level was undetectable. And she stayed on therapy for, it was either 15 to 18 months, and then was lost to follow-up and stopped antiretroviral therapy at that point. So didn't know what happened. And then mom and the baby resurfaced at 23 months. And at that time, the baby's HIV RNA level was negative. Her antibodies were negative. She had a single copy RNA done on her blood that was just barely positive at 1 copy. And her HIV DNA was copy number was 37. And they could not culture a virus from her blood, despite many millions of cells that they looked at. So remember, I told you that a lot of the DNA that we detect in proviral cells may be just garbage. So with a copy number of only 37 copies here, it's not sure that this represents real infectious HIV or if it's just junk. And then at 26 months, she again was HIV RNA negative, antibody negative. Her DNA copy number had now gone down to 4. And 2 LTR circles are measures of recent infection of cells, and those were negative. So everybody believes that this really is a profile of a child who is no longer HIV infected. And the question is, well, how the heck did that happen? And what's proposed is that infants really don't have any central memory cells. They haven't been around long enough to get any. And so there were no cells for her HIV to hide in, and therefore treating her for 15 or 18 months eliminated those first set of cells. Remember, the ones that are responsible for the most of the HIV replication in the plasma, and then that second order cells that are responsible for the second decline. Maybe those cells all died off, and those were the only cells in her body that were HIV infected, so that when she was released from HART at 18 months of therapy, there was no reservoir, and she is cured. So that's what everybody thinks. So what can we do for other people that aren't just born with HIV and unfortunately do have central memory T cells? That one strategy is to purge the reservoir. So in this strategy, this is a schematic of HIV hanging out in one of these central memory T cells that are pretty quiet, and there's not much HIV or any HIV replication going on. And can we wake it up? Can we stimulate it with HDAC inhibitors to open up the chromatin, IL-7, which is a way to get transcriptional factors available to the provirus, and prostratin signaling through protein kinase C, again, to upregulate some transcriptional factors, or DNA methylation inhibitors. Anything we can do to kind of wake up HIV in these resting cells so that it starts to produce viruses. And the hope then was that these activated cells would then die. They would either die because they're activated, which would stimulate a apoptotic pathway, and they would die, or they would be recognized now by the immune system that would kill it. And since the patients would be on HART, here HART is seen as a protective shield preventing infection of other cells, so that all you would be doing would be waking up the reservoir. The activated cells would eventually die, and that would purge the reservoir. The problem is that we do have medications that can wake it up, but it doesn't look like this step 2 death of these cells happens reliably. The little bit of data that we have from humans was published just last year by David Margolis's group out of UNC, and they were using an HDAC inhibitor, vorinostat, to get rid of the, or to keep deacetylation from happening and open up the chromatin structure to allow HIV to be replicated. These were 8 patients who were treated with vorinostat, and their resting T cells were then removed from their bodies and cultured ex vivo, and found that the patients that before and after they got vorinostat, there was evidence for much more HIV replication happening, kind of a proof of concept that we can give people these medications that will lead to upregulation of the HIV reservoir. So that's one strategy. A second would be therapeutic vaccination. This concept is to use an HIV vaccine to induce immune control of HIV in already infected patients, to kind of turn them into elite controllers. There's not any good human data that this has worked yet. In fact, there are human studies that were done using the Alvac vaccine against HIV. If anything, made things worse. They found that patients' T cells dropped more, and then kind of the state of activation induced by this vaccine led to declining CD4 cells rather than better control of HIV. But there has been one very interesting study done in rhesus macaques down in Oregon by Louis Picker and his group, and they used a rhesus CMV vector vaccine that had HIV proteins in it, then followed by an adenovirus 5 vaccine in SIV-infected animals. And 12 out of 24 animals developed complete control, maybe even eradication of their infection. And this is a little hard to see, I suspect, but I'll just walk you through it quickly. This is a DNA, and this is RNA, and these are animals. These are SIV uninfected animals, controlled animals, and this is 1, 2, 3, 4 monkeys that had the vaccination, and they had very little to no DNA or RNA in either lymph nodes or blood or other tissues. That's the difference, the red, the blue, the light blue, and the red. So they really couldn't find any HIV or DNA in any of those tissues in these 4 animals that was just like uninfected animals over here compared to infected animals that did not get the vaccines and had plenty of SIV detected in the tissue. So really remarkable level, and these were done at necroscopy. So they scrutinized these animals. They looked at every little tissue and found no evidence for persistent SIV after vaccination. So maybe we can replicate that in humans. A third strategy would be transplantation, and I'll just present to you some data from the Hutch about some patients who have been transplanted who had HIV. There's 2 flavors of transplantation. One is autologous transplant, where patients get TBI and chemotherapy to ablate lymphocytes, and then they are rescued with their own cells. So the good thing about that is that you don't get graft versus host disease. The bad thing about that is you might be giving them back their own HIV-infected cells. On the other hand, allogeneic transplant is transplant from somebody else. So you can use ablative or non-ablative, so conditioning regimens meaning just a little bit of chemo. So you end up with a chimeric person who's got the bone marrow of themselves and the bone marrow of the donor. This is obviously limited by donor histocompatibility. You can't just get a transplant from anybody. They have to be HLA matched to you. And the other downside is that you're going to get GVHD. That may be good because GVHD may be GVH against or graft versus HIV rather than graft versus host, but GVHD by itself also has significant morbidity and mortality. So this is some data from the Hutch. It's not very big data, but I thought I'd present it. So these were 3 patients who had HIV and another reason to get a transplant. One of them died and 2 of them were available for evaluation. This is those 2 patients. This is HIV RNA in these 2 graphs. This is HIV, infectious HIV in these 2 graphs, and this is proviral DNA in these 2 graphs. And the point is that after they got their transplants, there was a drop in the ability to culture HIV from their latent reservoir, and there was a drop in proviral DNA in that reservoir as well. It didn't get rid of it completely, but it did deplete it somewhat, suggesting that the chemotherapy used here had an effect on the latent reservoir. There have also been 4 patients at the Hutch who got an allogeneic transplant, so from a different donor, not from themselves. And they got non-ablative transplants, so they started out with chemotherapy that was not as potent as you'd get for an ablative transplant. And then when their immune systems reconstituted, they grew back their own immune system as well as that of the donor. But over time, their immune system became all donor. So this is the red line here. After transplant, this is mostly themselves, and then over time, this person has all donor cells making up their immune system. Same thing with this patient. Over time, it becomes complete chimera. Same thing here. And then this person tried to get the donor, and then the donor fell down, and they got most of their own bone marrow back. So not so good for their underlying lymphoma or leukemia. But you can see, as they went over to donor bone marrow, then they lost their proviral DNA. So in this case, maybe graft versus person is also graft versus reservoir. So, for what it's worth. So, the third strategy to cure HIV would be to engineer cells to eliminate genes, rendering them resistant to HIV, like the CCR5 knockout story, and I'll tell you about that in a minute. Targeting HIV provirus with gene-cleaving enzymes. The problem with that is that you would have to deliver these gene-cleaving enzymes to every HIV-infected cell in a person, which is not very practical. Or you could engineer cells by adding genes that render them resistant to HIV infection and then give them back to the patient. And then fourth would be combining gene therapy with transplantation to both deplete the reservoir and give them a new resistant bone marrow. So the way that most of the progress that's been made for gene-modifying therapy has been using these things called zinc finger endonucleases. This is a zinc finger right here. It's just a piece of nucleic acid that recognizes a particular sequence of nucleic acid, and you can design them to whatever sequence you want. So in this particular molecule here, there's 3 zinc fingers, 1, 2, 3, tagged to an endonuclease, so an enzyme that will cut DNA. And then there's 3 more zinc fingers over here tied to another endonuclease here. And if you choose your sequences correctly, then you can target any gene you want. And that's what was done here. They targeted a gene sequence of the CCR5 gene, and by getting this zinc finger to work on CCR5 genes, you will cut that gene here, and then it will recombine by homologous recombination, but it will be out of frame, and the CCR5 will not be expressed. So there's a few strategies here you can use. If we start with someone, and then you can bleed them and collect all of their cells that are CD4 positive and CCR5 positive. The little star is CCR5. And then you can introduce these zinc fingers that will cut that CCR5 out of some of the cells. And then give them back, expand them, so grow them up to billions, and then give them back to the person, and now some of the CD4 cells that this person would have would be CCR5 minus. Okay? So that's one strategy. Another strategy is to use that strategy with transplant. So here would be stem cell transplant, either from another person, so that would be allogeneic, or from the same person, that would be autologous. Again, you leukophorese them, but this time you select for stem cells, which are CD34 positive and CCR5 positive. You get all of those stem cells, and then you expose them to zinc fingers, so you knock out CCR5 in some of those stem cells, and then you give them back to the person, but in this case, you will have given the person chemotherapy or radiation to get rid of their own immune system, and then that would be an allogeneic transplant in this case, or if you took the cells from another person, it would be the first one was autologous, and then it would be allogeneic from a different person. But to give them back stem cells that were depleted of CCR5. So that was done and presented last year. There were a number of studies presented from Penn and then Sangamo, which is the company that makes these cells, back out in California with UCLA. There were 14 patients who were infused and 9 were evaluable at that time. They were given 10 to 20 billion zinc-modified cells, about 25% of which were CCR5 minus. And they found that in the patients who got these that their own CD4 cells rose over 100, and by day 90, there was still about 5% of the peripheral blood CD4 positive cells were CCR5 minus, suggesting that the cells not only survived in people by 3 months, but they were actually expanding a little bit. So this year's CROI, there was follow-up on that particular study. Again, this is adoptive immunotherapy. You're not treating the patients with chemotherapy or radiation, but again, using these zinc finger modified CD4 positive cells that no longer have CCR5 in them. 9 patients were infused with, again, 20 to 30 billion cells that they were expanded in the laboratory. And it again led to a median rise of 103 cells at 1 year. But they divided the patients into 2 groups of responders. There were high responders, 5 of them. They had their CD4 count increase by 227, and they also had their T central memory cells increase by 2.2%. These patients had lower levels of inflammation, which is defined as PD-1 expression, post-cell infusion, and also had lower levels of inflammation at the time that they got their cells. Whereas the low responders, and there were 4 of them, had a median T cell increase of only 44 and a median central memory increase of only 1%. And these people had higher markers of inflammation after cell infusion. So they were suggesting that this adoptive immunotherapy approach would work, but it's going to be important that we address the inflammatory state in HIV-infected patients. And if we can minimize that, then that would bode well for better survival of these modified cells. So what about gene therapy? Well, there's really no human gene therapy other than the adoptive immunotherapy I just told you, but there was an interesting paper that was presented from researchers here in Seattle at the Fred Hutch Cancer Center. This was stem cell transplant of pigtail macaques with a gene-modified stem cells expressing a fusion inhibitor that is called MC46 that makes cells resistant to, in this case, they were infecting the animals with SHIV. They had 4 monkeys, 2 were controls, and 2 got these gene-modified cells. And at 3 weeks, so these were monkeys that were treated with TBI after total body irradiation after or before they got the stem cells. So at 3 weeks after infection, these HIV-resistant cells here composed over 90% of T cells. So different than the adoptive immunotherapy, which only a few percent, because they're treating the monkeys with total body irradiation and they're giving them stem cells, 90% of their T cells at 3 weeks are HIV uninfectable. And these cells persisted over time. Their percent declined, but they declined not because their absolute numbers were declining, but they declined because the monkey's own CD4 cells were surviving, despite the fact that they were infected with SHIV, or SHIV is a combination of HIV and SIV, suggesting that the fact that they had HIV-resistant cells, these MC46 cells, were leading to better HIV responses that were controlling their viruses better, and that's what was leading to the higher CD4 count. So these monkeys had better HIV control, higher levels of anti-SHIV antibodies, higher levels of a CD8, CTL responses, as well as very robust anti-SHIV CD4 responses. So gene therapy that works, but not in humans, in primates. And then the last study that I'll talk about was an in vitro study. So we're going from humans to monkeys to cells here. This was another fusion peptide from GP41 that can prevent HIV infected. They took retroviral constructs encoding either of the 2 HIV receptors, CCR5 or CXCR4, and they fused at the N-termini with this new CD34 peptide. And what they found was that when they transduced cells with these constructs, that the cells were, even if the CD34 peptide was bound to the CCR5 co-receptor, not only did it block infection with CCR5 using viruses, it also blocked infection with CXCR4 using viruses. So it worked in trans. So you could put this molecule, this CD434 peptide, on either co-receptor, and it prevented infection not only on that co-receptor, but it prevented infection on the other co-receptor. So really, really quite remarkable. And then they did the same thing. This was in cell lines, and they did it in primary CD4 cells. And then they used, and it worked there as well, and then they used a zinc finger endonuclease to introduce the gene for that CD34 peptide directly into the genes for CXCR5 or CXCR4, and they found that when they did that, that those cells were also resistant to HIV. So pretty remarkable stuff here, that you could gene modify cells and render them resistant to HIV using this particular thing. So, but this again is only in cells and hasn't been moved into animal models or people yet. So, putting that all together, maybe the answer for curing people of HIV, if you're not going to get a bone marrow transplant like Timothy Brown, or if you're not going to get HIV within 30 hours of being born, that we can use these regimens that might be depleting regimens, and then adding HIV-resistant cells at various time points that might take a reservoir. Remember, I mentioned that the reservoir is only around 1 million cells, and if we could use some of these depleting treatments to knock that down, and on serial attempts here, maybe you could take a reservoir from 10 to the 6 cells and reduce it to only 10 to the 3 or maybe 10 to the 2 cells. And then if you've got people on fully suppressive antiretroviral therapy and you have a reservoir that's maybe only 100 cells, then maybe it does exhaust itself in a relatively short time frame, much less than the currently speculated 60 years. Maybe we can contract that to 3 or 4 or 5 years so that most people would have the prospect of cure. So that, in conclusion, I think we can say that HIV's cure is possible, either by eliminating the reservoir, that's what happened in the Berlin patient, or treating with HIV therapy before you even have a reservoir, that's probably what happened in the Mississippi baby. The reservoir is composed of long-lived cells that are central memory cells, but also some other candidates containing integrated copies of DNA, but only a fraction of that DNA is actually capable of producing infectious virions. The treatments, we can come up with treatments that activate the reservoir. They already exist, but the challenge will be to develop these therapies so that they're more effective, that they're not toxic, and that they not only activate the latent reservoir, but they actually kill those cells. And then strategies to use a functional cure using vaccines or gene therapy, introducing resistant cells, are promising and under development, but it probably will be a combination of reservoir depleting treatments with or without vaccines or gene-modified cells that really holds the promise for a sterilizing cure that we could scale to treat all people with HIV.